Hello and welcome to my video. My name is Manankumar Patel, and today I'm going to be giving you information on how cell cycle and cell signaling is related to cancer. Let me share my presentation. So, mm, cell cycle and cell signaling and its relationship to cancer. First of all, let's understand what cell cycle is and how does process of cell cycle look like. So the cell cycle is a process in which cell reproduce, mature, and eventually die at the end. This process includes several steps in it. I'm going to talk each of them later on in my presentation. So these are the 10 steps that are included in cell cycle. First is signal transduction, change in gene expression, activation of cyclin and CDK complexes, then phosphorylation of pocket proteins, DNA replication, passing S phase checkpoints, increase in cell size, then entry to M phase, then chromosome alignment during M phase, and lastly the cytokinesis. And as also I have mentioned the additional point for cell, when it dies in two different ways. First is necrosis and apoptosis. Apoptosis is like natural process in which cell divides into fragments, which is engulfed by phagocytes in the blood, and it is a natural one. And necrosis is like when you get an injury or some kind of cut, the cell dies by swelling of its organelles and leading to cell burst. So that is an additional point for you guys to understand. Let's move on to next slide. Okay. What is the first step of our cell cycle is signal transduction. In this, cell receives external signals that are required for cell growth or cell division. CDKs acts as regulators which make sure that there are favorable conditions for cell to advance to next phase. Let's move on to step 2, where there is change in gene expression. When cell reaches the restriction point, it can't rely on external factors anymore, and it passes through this point. Cell produces protein called cyclins, and these cyclins team up with CDK, forming complexes that help the cell to pass to next step, or you can say next phase also. See what step 3 is. Step 3 is of activation of this protein produced by cell, which are cyclin and CDK complexes at G1 and S checkpoint, which is the first checkpoint that cell has to pass. When the cell reaches the end of G1 phase and is about to enter S phase for DNA replication, it has to pass the checkpoint. It is where cyclin and CDK proteins play a role in helping cell to signal to move forward by forming complexes of cyclin CDK complex, which is called activation. There are two signals called pro-growth and anti-growth, which indicates if cell goes further or not. If pro-growth signals are stronger, then cell moves forward, and if anti-growth signal are stronger, then cell will stop and not move forward. As you can see on the image also that there is a G1 phase and checkpoint is a G1 S phase. That is the red symbol. As you can, CDK2 and cyclin E will form a complex over there for activation. And moving on to step 4, which is a phosphorylation of pocket protein, also known as E2F. This process also happens at same checkpoint. This cyclin and CDK complex will do phosphorylation of pocket proteins, which will release E2F, which will allow E2F to activate genes required for DNA replication in S phase. Step 5 is the DNA replication through protein kinases that happens in S phase. Before cell starts copying DNA, it requires help of protein kinases, which act as switch for machinery that activates DNA replication. There are certain cyclin and CDK complexes combination which act as a master switch for replication process. These complexes activate other proteins required for replication such as helicase and DNA polymerases. Now, after the DNA has replicated, it has to pass through S phase checkpoint at the end of S phase, which is S G2 checkpoint. So, which ensures this checkpoint ensures that DNA, that new DNA that is formed is correct and there are no mistakes. Then there is G2M checkpoint, which is final checkpoint before cell divides. It verifies that DNA is intact and properly copied. If there is a DNA damage, then cell will stop forwarding and try to repair it. If it has like major damage which cannot be repaired, then cell will perform apoptosis, which is cell program death. The cell will eventually die itself. Now move on to our step 7, which is increase in cell size, G2 phase. Now cell enters G2 phase where it continues to increase its size to ensure that it has enough materials for two cells. It starts to form new copies of its organelles and other parts so that the cell will have everything it needs to function properly. It begins storing energy which will require later for dividing into two cells. At last, cell is scanned at G2M checkpoint before it moves to M phase. Now, the cell, after passing the checkpoint G2M, which is final before the DNA, after the DNA replication, the cell will enter M phase, where now dividing of cell into two will begin. Here, cyclin B and CDK1 will form complex and act as a switch for initiation of cell division. Moreover, other proteins are activated which are necessary for chromosome condensation, nuclear envelope breakdown, and spindle formation. M phase has four phases in it, which are prophase, metaphase, anaphase, and telophase, which will in prophase the chromosome will align at the center, and then in metaphase they will the spindle will touch them, and in anaphase they will try to pull apart on the each poles, and in telophase the nucleus will be divided into two daughter nuclei. These are the four phases that happens inside M phase. Now moving on to our next, where chromosome alignment during M phase. So as I was talking, first phase is prophase, where cell DNA condenses into two identical chromosomes known as sister chromatids. Second is metaphase, where chromosome lined up in the middle of cell. In anaphase, the spindle fiber are fully formed and plays role in separating the chromatids by pulling them apart towards opposite poles. The alignment check is performed to ensure that all chromosomes are aligned and attached properly to spindle fiber. Then at last, in cell cycle, cytokinesis take place. This is where rest of the cell, including cytoplasm, needs to divide to make sure that new cell gets everything in it to function. Then each new cell can run and perform their own cell cycle. Now we'll understand what cell signaling is, which is a part of cell cycle also. Cell signaling is a crucial part of cell function which helps them to communicate and coordinate between among themselves. It is important process like cell growth, immune response, and homeostasis. The components that are involved in signaling are signal receptor transduction mechanism, or also known as transduction pathway, and a cellular response. External signal are transmitted to internal components of cell, and the purpose of the signal transduction pathway is to convert the extracellular signal that was received by receptor will be converted into a cellular response, which signals internal components to function. We will understand the structure of signal transduction pathway. How does the pathway work? First, the signal is initiated by signaling molecule, as you can see in the figure. It can be, there are various type of signaling molecule that I'll discuss further also, which is detected by receptor on the cell membrane of cell, and then the transduction happens is the process where the signal is converted to cellular response using transcription factor, and later on the response is created. Now, as I was saying, types of signal and receptors. So there are several types of signal, such as physical signals, then hormones, neurotransmitters, growth factor, etc. Receptors are protein where signaling molecule bind and initiate response. So there are six different types of receptor with different functions. Uh, here I will talk about just two, such as G protein coupled receptor and tyrosine kinase receptor. And we'll understand what this receptor pathway function individually. Now, next slide. So first we'll see how G protein coupled receptor is involved in a signaling pathway. Here, a signaling molecule binds to an extracellular domain of GPCR. This binding causes conformational changes to GPCR. So now this altered GPCR interacts with nearby G protein, converting GDP to GTP on G protein alpha subunit. Now this G protein subunit will divide up into three parts: alpha, beta, and gamma subunit, which interacts with other intracellular targets to transmit the signal. This interaction leads to production of second messengers like CAMP, which further amplifies this signal to other intracellular targets. Now, we'll understand tyrosine kinase receptor, how they work in signaling pathway. A growth factor, FGF, and or hormone, there are several other types of growth factor, binds to extracellular domain of FGR, which is a FGF receptor. FGFR is a type of RTK, like tyrosine kinase receptor. This binding leads to dimerization of FGFR. These dimerized FGFR phosphorylate each other on specific tyrosine residue process known as autophosphorylation. This phosphorylated tyrosine serves as docking site for other various intracellular signaling proteins. Eventually, this protein activates downstream signaling pathways such as MAP kinase pathway. Now, we'll discuss how cell cycle and cell signaling is related to cancer. First, let's see how dysregulated cell signaling leads to cancer. Um, what cancer is cancer is a disease when mutation or changes uh, in signaling pathway disrupt the normal cell process. As we discussed, cell signaling includes pathway that regulate cell behavior. Each pathway disruption leads to different cancer type. For example, overactivation of ATK pathway is common for breast and prostate cancer. Then WNT pathway is related to colon cancer. Then MAPK pathway is related to colorectal cancer and melanoma also. Then overactivation of this growth signaling pathway leads to mutation in gene like EGFR, which causes constant activation of signaling pathway. So there is no stopping for cell division, and cell will multiply uncontrollably, causing tumor formation and leads to cancer eventually. Then cancer cell are clever, so they inhibits immune response and avoid detection by making them think they are body's own cell. So the immune response will not act against cancer cell, and the tumor will spread among the different parts of body. Now see how cell cycle is related to cancer. As we learned earlier, that cell passes through several checkpoints before entering for final division. Disruption in cell cycle is primary factor in development of cancer disease. It is like the basic uh, point where the cancer starts forming in the body. The first checkpoint cell passes is G1S, which is at the end of G1 phase, where cell is about to enter S, where even if DNA is damaged, still bypasses and proceed to G2M checkpoint. This happens when the cancer has formed. There is a protein called retinoblastoma protein which regulates G1S checkpoint, and mutation of that protein leads to uncontrollable entry into the cell cycle. That's how the cell bypasses each checkpoint for like this checkpoint, G1S checkpoint, due to mutation in retinoblastoma protein, RB protein. Overactivation of cyclins and CDK, which are responsible for verification of cell before it passes checkpoint, leads to unchecked progression of cell through each step of cell cycle. So these were the activation of cell cycle, CDK and CDK complexes. So uh, there is a mutation or overactivation of these proteins will lead to cell progression without unchecked for each checkpoint. At G2M checkpoint, where DNA is checked for any damage, and if found, then it performs apoptosis. So when this apoptosis pathway is disrupted, it will allow cell to evade program cell death. So that's how cell will escape apoptosis by duping this G2M checkpoint. And let's see. For concluding my topics overview, I would say cell cycle and cell signaling are primary processes which make sure proper cell growth, division, and survival. Cancer is major disease that is caused by malfunction in cell cycle along with cell signaling. Currently, various ongoing research is conducted for developing treatment for cancer using method that target specific tumor suppressing inhibitors such as CDK inhibitor, PRAP inhibitors, etc., and many more. Relationship between these two has helped advancement in medicine such as targeted therapies and immunotherapy, giving hope to patients that some day in future they will develop cure for cancer. Also, deep research about intracellular mechanism needs to be done to understand more about cell signaling pathways so that specific treatment can be made. So that is my point of view for like understanding more deep about the cellular mechanisms that happens inside cell during cell cycle or cell signaling, so that you can understand more far better how to develop or how to stop cells from uh, stopping immune response when they are like duping apoptosis at the end of cell cycle. So that's all. I hope you like my video, and you by the end of this like my presentation you should have gained some idea what cell cycle is and cell signaling and how the disruption in them can cause or lead to cancer like disease. So thank you. 19 minutes.